All right, welcome. Today, we're diving deep into the key virology and clinical management of human papillomavirus, and we're going to be looking at it specifically through the lens of the Dutch context. So this guide is really tailored for you, the medical microbiologists out there. And just so you know, all the information we've pulled together for this was compiled using Gemini Deep Search. If you want to see the full list of sources, you can check out the linked Notebook LM.
So, here's our game plan. We're going to follow a classic bench-to-bedside arc. We'll kick things off with the virus itself, then get into its clinical impact and how we actually spot it in the lab, and we'll wrap up with treatment guidelines and, of course, the Dutch prevention strategy.
All right, Section 1. Let's get right into it and start by profiling our subject, the virus.
So here it is, human papillomavirus. You probably know it as the most common sexually transmitted infection on the planet. But for our purposes, what's really crucial is that it's the necessary etiologic agent for several cancers, with cervical carcinoma being the big one, of course.
Okay, let's talk structure. What we're looking at here is a non-enveloped particle, and it's tiny, about 55 to 60 nanometers across. The key thing to notice is that it has this incredibly stable icosahedral capsid, and that stability, that's what makes this virus so darn resilient out in the environment.
Now, this is where it gets really interesting from a structural standpoint. Take a look at that capsid. It has this unique construction. It's built exclusively from 72 pentameric capsomers. Think of it as an all-pentamer build. This is actually pretty unusual for its symmetry class, a T=7 lattice, because you'd normally expect to see a mix of pentamers and hexamers. So it's a real structural outlier.
Okay, so where does it fit in the grand scheme of things? Taxonomically, it's part of the Papillomaviridae family. But the one you really need to remember, the genus that's most clinically significant for us, is alpha papillomavirus. Why? Well, because this is the group that contains the whole spectrum, from the low-risk types that cause warts all the way to those high-risk types that are linked to cancer.
Okay, let's shift gears now from the virus to the patient. What's the actual clinical impact? How does this virus show up?
The clinical spectrum for a human papillomavirus is just incredibly broad, and it really all depends on the type. This distinction is absolutely critical when it comes to diagnostics. You've got your low-risk types, like 6 and 11, which mainly cause benign stuff like anogenital warts. But then, you have the persistent infections with high-risk types. We're talking particularly about 16 and 18, and that's the primary driver of malignancy.
So how does it actually cause cancer? Well, the linchpin for malignancy is the continuous expression of two viral oncoproteins, E6 and E7. Think of them as a tag team. E6 goes after and degrades the tumor suppressor p53. Meanwhile, E7 inactivates another key player, the retinoblastoma protein, or PRB. So together, they just completely dismantle the cell's main defenses against uncontrolled growth. It's a really effective and sinister strategy.
All right, on to the lab. This is where we, as microbiologists, play such a central role in managing this virus.
There's a fundamental challenge in working with human papillomavirus, and it's one that has shaped our entire diagnostic approach for decades, and it's this. You simply cannot grow it in a standard, conventional, monolayer cell culture. The reason is that its entire life cycle is completely tied to the differentiation process of its host cell, the keratinocyte. No differentiation, no virus replication. It's that simple.
So, because you couldn't culture the virus, what did that lead to? Well, it led to a total reliance on morphology, literally just looking at cells under a microscope. For years, the cornerstone of diagnosis was spotting these specific cells called koilocytes in pap smears. These became the pathognomonic sign, the dead giveaway of a productive infection. Seeing one of those was the classic hallmark.
And that brings us to today, which is a whole different world. This table really shows that evolution from just looking at cells to using these powerful molecular methods. DNA PCR, that's the gold standard now for primary screening here in the Netherlands. But RNA-based assays, they take it a step further. They don't just tell you if the virus is there, they tell you if it's transcriptionally active by spotting that E6/E7 messenger RNA. That gives you much higher specificity for the lesions that actually matter. And on the horizon, we have things like ctDNA analysis, which is this incredible emerging tool for monitoring treatment without invasive procedures.
So, we found the virus in the lab. Now, let's head over to the clinic and see how those diagnostic findings actually translate into treatment strategies.
Okay, this is probably the most critical point when we talk about treatment, so it's really important to get this straight.
There is currently no systemic antiviral therapy that can eliminate the human papillomavirus infection itself. Let me say that again. We can't cure the virus. All the treatments we have are aimed at the lesions, the warts, the CIN, not the underlying viral infection hiding out in those basal epithelial cells. We're treating the symptom, not the cause.
And this is where the Dutch context becomes really interesting. For something like a CIN2 lesion, where we know spontaneous regression is pretty common, the Dutch guidelines take a much more nuanced approach. Instead of jumping right to excision, there's a big emphasis on active surveillance, or maybe uterus-sparing options like topical Imiquimod mud. This is especially true for women who might want to have children later. It's all about this careful balancing act, weighing the risk of progression against the real iatrogenic risks of treatment, like premature birth, which you might see with a more interventionist first-line approach that's common internationally.
Right, so that begs a pretty big question, doesn't it? If we don't have a direct antiviral to get rid of it, how on earth does this virus manage to stick around for years, evading our own immune system?
Well, the answer is that this virus is an absolute master of disguise. It has this incredibly effective, highly evolved strategy of immune evasion. It's this ability to hide from and actively manipulate our immune system that allows it to persist for so long. And that persistence, that's the #1 risk factor for developing cancer.
And it's got a whole bag of tricks to pull this off. First, its entire life cycle is completely hidden away within the epithelium. It doesn't cause cell lysis, there's no viremia, so it never really sounds the alarm. And second, remember those oncoproteins E6 and E7? Well, they're back. They actively disrupt our innate interferon response and, get this, they suppress antigen presentation on MHC class I molecules. That essentially makes the infected cells invisible to our cytotoxic T cells. It's like they're wearing an invisibility cloak. All of this, the virology, the persistence, it brings us to the bigger picture, the public health strategy for controlling human papillomavirus in the Netherlands. And it really is a fantastic case study. You know, the Dutch strategy is truly a world-class example of integrated prevention. It's built on two incredibly powerful pillars. You have primary prevention, which is the National Immunization Program, and you have secondary prevention, the Cervical Cancer Screening Program.
And let's look closer at that secondary prevention pillar, because the screening program is really advanced. Since 2017, the whole thing has been based on primary high-risk human papillomavirus molecular testing. But it also includes some fantastic innovations, like offering self-sampling kits to lower the barrier to participation. And it's smart. It uses risk stratification to optimize how often people get screened, which means we can safely extend those intervals for low-risk individuals.
But here's the thing. Looking ahead, the incredible success of these two programs working together actually presents us with our next big challenge. As more and more vaccinated people enter the screening age, the prevalence of high-risk human papillomavirus is going to plummet. That's great news, obviously, but it also affects the predictive value of our tests. So what that means is we're going to have to be clever and adapt to optimize the screening algorithms for this new, vaccinated reality.
So we hope this deep dive was valuable for your work. If you enjoyed this, please do subscribe for more content that's tailored specifically for medical microbiologists. And we'd really love to hear from you. We truly value your expert insights, so jump into the comments and let us know, what do you think is the next big step for human papillomavirus screening algorithms in these vaccinated populations? We're really looking forward to reading your thoughts and getting a great discussion going.